Hi, my name is Sophia Felbaum, and for my senior research thesis, I conducted an experiment on fungal succession in rat cadavers. To introduce my project, I'll first begin talking about forensic mycology. Forensic mycology is a relatively new field that is useful in forensic investigations. It combines the use of forensic science and as well as the background of mycology in identifying several factors in criminal investigations. Forensic mycologists combine the knowledge of mycology with criminology by using fungal data that can aid in estimating the post-mortem time interval, determining the gravesite of a corpse, and even using it as trace evidence. Fungus can exist in numerous types of environments, ranging from outside on the ground of a forest to the inside of a shower. Since fungal species are varied and are found on a wide range of places, they can be found in cadavers either from growing on the corpse with different species competing for succession or from spores being found on the clothes to identify where the body was originally. Postmortem changes is whenever a corpse naturally decomposes and changes at a cellular level. Not all corpses decompose the same way, however, and so different factors influence how it decomposes, such as temperature, location of the corpse geographically, the weather, and just in general the environment. Whenever they're taken into account, an understanding can be made of how a corpse decomposed in the way that it did, and this information can be utilized to determine when and where a corpse came from without any prior knowledge about it. There are some immediate changes after postmortem when an individual's vital functions have ceased, and in particular the functions of the nervous, respiratory, and circulatory. Early postmortem changes are typically identified as the initial breakdown of soft tissues of a body, and these stages are algor mortis, livor mortis, and rigor mortis. Some of the stages of decomposition are in 5 known stages, starting with fresh, bloated, active decay, advanced decay, and skeletal. The fresh decay stage is whenever autolysis is occurring and early postmortem changes can be identified. Insects like blowflies and flesh flies are the first insects that are attracted to a corpse and are typically of an indicator of the end of a fresh stage, and the life cycle of these insects coincide with the bloat stage. During the bloat stage, the putrefactive gases accumulate within a corpse, causing it to swell. The buildup of these gases are due to the microbiome of the corpse. The anaerobic activity of the bacteria inside exerts pressure on the body up to the point where it bursts. Active decay is the acceleration of putrefaction, and the bodily fluids from the process are otherwise forced out. Advanced decay is the stage where the bones of the corpse are exposed, and the corpse kind of caves in. This stage is also called black putrefaction or late decay. The skeletal stage is when the bones of a corpse are mostly exposed, and dry skin, tendons, and cartilage are in minimal amounts. This is also considered the dry stage or skeletonization. Moving on to the necrobiome, it is similar to how we each have a microbiome within our bodies, except it's a microbiome that is involved in the decomposition process. And so it describes prokaryotic and eukaryotic species that are part of decomposing of the mass remains. Microbes contribute to a wide variety of biological and ecological processes. One of those ways is through nutrient cycling into the environment in the process of decomposition. Around 2 billion metric tons of high nutrient animal biomass contribute to the ecosystem through productivity and soil fertility. Nutrients that come from the biomass of carrions go into the environment for microbes metabolizing it into the surrounding soils, where it can be consumed by scavengers that are vertebrates or invertebrates. The decomposition of animals differs from plants in that proteins and lipids are primarily broken down in a different metabolic process. Microbial communities may come together at certain sites and try to outcompete other existing communities of microbes for nutrients on a corpse or the surrounding area of it. Microbial communities are affected by several factors, which include the mass of the corpse, the pH of the soil, temperature of the environment, humidity and moisture of the surrounding area, and water activity. Fungi are a part of that microbial community, and they also aid in the breakdown of organic matter. A variety of fungal members occur with bacteria in these processes. Due to fungi metabolizing organic matter in one place, they alter biochemical and physiochemical properties in the ecosystem, as well as the microbial community structure. A variety of fungal species can colonize around a cadaver, but the most common species that have been identified to do so are the genera Aspergillus, Penicillium, Candida, and Mucor. These species typically appear in the bloat, putrefaction, and skeletonization stages of decomposition. And when a cadaver decomposes and the tissues are broken down, the fluids are leaked out. A complex nutrient pool changes the surrounding environment in a way that provides nutrients for the nearby plant life. The cadaver has an effect on the environment where the chemical composition of the surrounding soil is altered. In the process of decomposition, a corpse will release different compounds of varying diversity, those being adipose tissues, fatty acids, and organic nitrogen. Biogeochemical cycling is enhanced into the soil below a cadaver. Studies have shown that there is an increase in microbial respiration rate and nitrogen mineralization. Soil microbes themselves are critical in mineralization of the primary production of terrestrial ecosystems. Post-putrefaction fungi or ammonia fungi undergo a succession where dominant fungi can be taken over by another kind. Succession can only occur if the previous fungal species dies. Fungi that develop in the early stages typically fruit on soils that are highly concentrated in NH3. Fungal succession seen in cadavers is possibly similar to the fungal succession of ectomycorrhizal fungi that develop in the forest. Specific fungal populations have been characterized into ecological niches that are well defined. And overall, for my experiment, I have come up with 3 hypotheses that I wanted to test. The first being the rates of decomposition between indoor and outdoor rat cadavers. The second being the fungal succession indoors and outdoors. And the effects of soil pH on fungal succession. And I was hoping to learn more about fungal succession and how it has an effect on cadavers. The methods for my project begins with prepping the indoor and outdoor rat cadavers for the experiment. Starting with the outdoor rat cadavers, I went to the designated area where they were to be left to decompose, and I tilled up the soil so that there wasn't any grass or vegetation in the way, and the rats could just sit on the soil to decompose. And I made it about a foot wide in diameter, and I spaced them out evenly, about 2-3 feet away from one another, in 6 spots. There would be 2 rats decomposing on basic soil, 2 rats on acidic soil, and 2 rats on unaltered soil. I would till in soil acidifier and lime to either make it more acidic or more basic. The lime making it basic, the acidifier making it more acidic. I would use 30 grams of each for each soil location. I then placed the rat cadavers in the center of the tilled soil spot, and then I placed a metal basket over each rat cadaver and put metal garden stakes on either side of the basket and hammered it in with a mallet so that way scavengers and large animals couldn't get the rats and they could just stay there. However, they were still accessible to small scavengers like the outdoor insects and microbes. The indoor rats followed a similar procedure, except they were in Tupperware containers, and so I took soil from the same location that the outdoor cadavers were in, around that area, and I put about 1 to 2 inches of soil into each Tupperware container. So there were 6 containers, each with 2 basic rat soils, 2 acidic rat soils, and 2 unaltered. Once I added 30 grams of each of the soil acidifiers or lime, I placed the rats in the middle of the soil, in the middle of the Tupperware container, and I closed the lids on each of them. I stabbed holes into the lids of the containers using a knife. So whenever I had the dermestid beetles, I would be able to add them in and they would have breathing holes to help speed up the decomposition process, because it would take a lot longer for the indoor cadavers to decompose than the outdoor ones, since they don't have as much access to scavengers and decomposers as the outdoor ones do. I then sterilized my materials and made plates for my project. And so I had my medium, which was SMA or sorbode maltose agar. I put 65 grams of the agar powder into 1 liter of deionized water that was brought to a boil. I would then put it into the autoclave, and then once it was done autoclaving, I would take it out and pour it into petri dishes while using a heated glove to hold the flask by the neck. And then using my other hand, I would open the petri dishes and pour them out, and then close the petri dish to keep it from being contaminated. I would then let them cool on the lab bench after I had used all the agar in the flask. Sometimes I would use more than one flask to make a lot of batches of plates. Once I had made the plates, I would go collect the samples from the cadavers on the days that they were designated to have the samples collected from them. And I would take sterile swabs that were also autoclaved, and I would swab the underneath or the underside of each rat cadaver, indoor, outdoor, just where they kind of made contact or close to where they made contact to the soil. If the sterile swabs were in their own little packaging, I would put the swab back into the packaging. I would carry it inside, and then I would go into the lab and I would make my dilutions with the samples. And so I had centrifuge tubes that I used that each had saline solution, and I would make my dilutions. I first began with dilutions that ranged from 10 to the -1st to 10 to the -3rd. And so there was 3 dilutions per rat, and then there would be 3 plates per dilution. So each rat cadaver had 9 plates made for them. And then eventually, throughout the semester, an increase in colony count occurred, and so I had to do more of a dilution, and so I had to do 10 to the -5th to 10 to the -7th later on in the semester, because the colony counts were too numerous to count. After I had made the plates, I would put them into an incubator where it was set to 30 degrees Celsius. And then after 24 hours of being in the incubator, I would take them out and I would count them, and then I would put them back into the incubator, and then a week later I would check those same plates and count them again. And then after I had counted everything and my project was all over by December, I used a repeated measures analysis to analyze my data. When looking at the fungal colony counts at 24 hours of incubation, using a repeated measures analysis did not show significant data or effects with the soil treatment, dilution, air temperature, carcass age, or indoor versus outdoor cadavers. The trends that weren't significant showed that the second basic soil treatment, or B2 in Table 1, had a similar fungal colony count of 3.3 as the controls did, which shown in the table are from 3.2 to 4.7, and the controls are the stars 1 and stars 2. Both of the acidic soil treatments, A1 and A2, and the basic soil treatment, B1, had lower colony counts with least square means of 0.6, 0.4, and 0.9 respectively. The data can be found in Table 1 as mentioned previously. And then looking at Table 2, comparing the counts of the indoor rat cadavers to the outdoor ones, there wasn't much of a difference between them, with the indoor being 0.7 and the outdoor 3.4. When looking at the fungal colony counts at 1 week of incubation into consideration, the repeated measures analysis did show significant trends and a variety of effects. The acidic soil treatment A2 shown in Table 3 comparatively produced a lower fungal colony count of 2.4 compared to the other soil treatments, which ranged from 11.3 to 29.4. Oppositely, the basic soil treatment B2 had a higher fungal colony count of 29.4 than the other treatments, which ranged from 2.4 to 18.4. Data of this can be found, as previously mentioned, in Table 3. In looking at Table 4, comparing the indoor rat cadavers to the outdoor rats, the least square means table shows marginal difference in counts, where the indoor cadavers were 58.3 and the outdoor was 0. Based on the results of this experiment, it would seem that the colony counts were most significant at 1 week of incubation instead of the 24-hour mark. After 24 hours of incubation, initial colonies would form, and so likely the fungal colonies weren't able to develop to the full extent that they could compared to 1 week of incubation. Due to varied species of fungi growing and not identifying them to their specific species, and just using colony morphology to identify them, their incubation times likely varied and proved more beneficial to them to be able to grow for a week's time. A variety of factors could have influenced the experiment overall as well, one factor being contamination. The agar used in this research was sourbrod maltose agar, which is best used for yeast and fungi. However, it is not the only microbe that will grow in the medium. Certain types of bacteria will also grow in the medium, and more specifically bacteria that will tolerate a low pH environment. There was some growth that appeared on the plates that looked related to bacterial colonies. However, specific species identification proves to be difficult for this research. To identify specific fungi and bacteria, more types of microbial testing would have been done instead of just identifying the colony morphology. It is also possible that microbes from other sources other than the cadavers and the soil grow on the medium. It's less surprising with the outdoor cadavers as they are exposed to the elements in different scavengers, but the indoor cadavers are in a more controlled environment, so this would be more of a concern for contamination. Results may have been varied for the outdoor rat cadavers due to the climate changing over the course of the fall semester in 2024. The project has started with the indoor cadavers on October 17th and ended with the outdoor cadavers on December 10th. Over the course of 3 months, the outdoor climate had become colder and even snowed some days. The outdoor cadavers toward the end of the experiment had been frozen and showed little colony growth. Had the project been done over the summer, results likely would have been different. However, the project could use some more research during different seasons to gauge the effects of weather patterns on the cadavers. The indoor cadavers had reached a stage of decomposition where their bones were showing because of the domestic beetles helping to speed along their decomposition process. However, the outdoor ones were only able to mildly decompose. Their bones had not even showed by the time that the experiment was over. A larger sample size would have been ideal to collect data from, with more rat cadavers helping to be more accurate. More samples taken could have shown extensive data over the course of not just 24 hours and 1 week of incubation time, but perhaps 48 hours, 72, etc. As mentioned previously, gathering data from the cadavers throughout the year would prove to be useful in finding changes in fungal species in not just the cadavers decomposing, but also seasonally. Unfortunately, only so much can be done with the resources and time given, and so the research is done within the confines of what is possible with what is had. The hypotheses tested was the rate of decomposition in an outdoor environment and a controlled indoor environment, fungal succession in both, and the effects of soil pH on fungal succession and the rate of decomposition. The predictions that were made were that the outdoor rat cadavers would decompose at a faster rate than the indoor cadavers. The fungal succession of this species will differ between the indoor and outdoor cadavers, and fungal species will do better in soils with a lower pH. The rate of decomposition did vary between the indoor and outdoor cadavers. However, as previously mentioned, the indoor ones did decompose faster than the outdoor ones due to the weather having freezed the outdoor rats. Similar fungal species could be noted by the colony morphology between the two, but there was notably different fungal colonies between on either of their plates. The acidic soil treatments seem to have less counts than either the controls or the basic soil treatment. Of the hypotheses tested, at least one of them was proven to be correct. However, it will still be informative in learning the outcome regardless of whether the hypotheses were proven to be correct or not. Overall, I think this research was important in helping out in understanding forensic mycology and mycology as a whole, and they're both very interesting fields to learn about, and so with more development and research, more uses can be made of them in the future.